Hello, welcome to my point mutations video. In this video, we're going to talk about different types of mutations like frame shifts and non-frame shifts, and I want you to understand the difference between those different types of mutations like addition and deletion, substitution, inversion, and all of that good stuff. So, different things that can happen during DNA replication that can cause a mutation. We're specifically just talking about letter changes in this video. We're going to have a separate video talking about what could happen during chromosomal mutations. So, totally different video. So first, DNA polymerase is the one that has to add the letters. Sometimes it will add the wrong letter, and yes, its job is to proofread and fix any mistakes, but DNA polymerase actually kind of sucks at its job, so it does sometimes leave mistakes in our DNA. So this is called a point mutation. Depending on the type of mutation, if there a letter's added, a letter's taken away, if we add in or we invert a couple letters, different things can happen. But once it's there, the next time the DNA replicates, it will replicate it the same and continue replicating it every time the DNA is replicated. So every time we do cell division, that DNA mutation now is there forever because the DNA polymerase isn't going to know that three replications ago it got wrecked. So it's just going to read it as it's supposed to be there. So in that case, proteins, like for example, if it was a cell in the pancreas that creates insulin, that protein maybe isn't made correctly anymore. So it is really important that DNA polymerase does this correctly, but sometimes we do get left with mistakes. If we expose our body to something called a mutagen, this can cause DNA replication to not work as well. So mutagens are things like radiation, for example. So things that we expose our body to that can increase the chance of these mutations to happen. So just so we know for later, I'm not going to talk a ton about this because you guys don't know what protein synthesis is, but we know that A's and T's and G's and C's, they are all in triplets of DNA. So our DNA is written in 3-letter words called triplets. So when we are reading our sentences, they're always 3-letter words like ACT, space, GGC, okay? So our DNA will read like that. If our DNA gets messed up, sometimes this process can then wreck those triplets and then wreck what it codes for. So the specific amino acid that will then become a protein could potentially wreck that one amino acid. And if that one amino acid is wrong in our chain of protein, that protein will be all messed up. So we're going to talk about 4 different types. So we have frame shift and non-frame shift, but that includes additions and deletions, substitutions, and inversions. So we're going to talk about those 2 different types of each type. So 4 types in total. So first, let's talk about frame shift mutations. So in this case, every single triplet of DNA is going to be affected after the mutation occurs. So first, I want to give you an example that we're going to be using throughout the video. The word beast, okay, from Beauty and the Beast. So the word beast is our original word. If we had an addition, it would be like adding a letter to beast. Okay, so for example, if I added an R after the letter B, it would make the word breast. Breast is not the same as beast. It means a totally different thing. So when you're reading the word breast, you don't think at all like, oh, maybe they meant to say beast. Okay, it's kind of like decoding a text message with a lot of typos. Okay, your cells aren't very good at doing that, so they would be very confused if it said this. But not only that, if you had a sentence after the word beast, but you added in an R, and it said breast, everything after would get messed up. Okay, because our DNA stays in letters of 3 and 3 and 3, the spaces have to stay in the same spot after every 3rd letter. So the entire sentence gets all jumbled up. So in our actual DNA strand, it would look like this. So maybe our original is AAC, TCT, ACC, and maybe that's the part of the DNA that codes for insulin. Well, now all of a sudden, if we add a letter into this, all of those spaces stay in the same spot, like I said, but everything is shifted down. So now when our body's trying to read the instructions on, hey, this is how you make insulin, it now no longer makes sense because it looks totally different. Now for us looking at this, it's hard to kind of understand because we don't read DNA language. But if this is in English and the sentence now had a bunch of jumbled words like breast instead of beast, it would be really hard for us to read the instructions, just like it is for our cell. The other type of frame shift mutation that we can have is a deletion. So if we go back to our word beast, our original word, if we had a deletion, it would be like taking away a letter. So for example, if we take away the A and now it says best, best and beast mean two different things. So if we took from our original DNA strand, again, let's just say this is the one that codes for insulin, and we took away a letter, again, it's not going to code for insulin. But also everything after that in our DNA, all of the other genes, because we took away one letter, everything shifts to fill that space. We won't just have a space in our DNA. All of the nucleotides will shift up to fill that space in our DNA, and literally everything after that space will not make any sense whatsoever. So that is why frame shift DNA mutations are so dangerous. They literally will destroy all of your DNA, basically, especially if the DNA mutation happens at the very beginning of your DNA. Everything is destroyed after. Your cell will most likely not be able to survive if a frame shift mutation happened. Okay, so not only does it mess up the insulin gene, but it would mess up every other gene in your DNA after that mutation. It's super, super, super bad if this happens. The cell probably will not survive. They won't be able to read any of the instructions. The other type are a little bit less dangerous, so non-frame shifts are ones like and substitution, where we just take a letter and substitute it with a different letter. So for example, if we took a B and substitute it with an F, now it says feast, which still says something different, but at least it doesn't mess up everything after that. Okay, so still dangerous, but maybe now only the insulin gene is messed up. So that person now has diabetes or diabetes-like symptoms, but they can still make all of the other DNA genes and all the other proteins in their body. Okay, still not great, but it also is interesting that sometimes frame shift mutations will happen and you still end up actually coding for the same amino acid, because amino acids actually have multiple different combinations of letters that can mean the same thing. So for example, when we say there, there's actually 3 different spellings of them, but they still sound the same. So we can kind of figure out what that person means, even if they use the wrong there, even though it's like grammatically incorrect, we can still figure out what they mean and still read the instructions. So that's kind of like a frame shift mutation. Sometimes it will actually mess up and code for the wrong amino acid, and that will mess up that gene, but other times it will make, even though it changes the letter slightly, it will still actually end up coding, because to our cells, it still makes the same amino acid, because amino acids can have multiple spellings for that triplet of DNA. So yeah, basically what I just said out loud right here. Unique thing about amino acid triplets is that there's a lot of different like options of things that they could write to be serine still, which is an amino acid. And then the last type of non-frame shift that we have is an inversion, so taking letters and just reversing them. So if we take the S and the T in beast and switch them, it now says beats instead of beast. So again, a different word, and then we have an inversion in this. We have it bolded. This is the one that inverts, okay? Again, inversions and substitutions are usually not as harmful, because they might only harm one of the actual genes or one of the proteins, or sometimes it actually ends up not even affecting it at all. Like you would never know that it was a mutation at all, which is pretty cool, because sometimes there's multiple spellings for the same amino acid, which is pretty awesome. Okay, if that happens, that's like super lucky, but at least it's only, if it does happen and causing a mutation, at least it's only messing up one gene instead of literally everything in your DNA after where the mutation occurs, because everything shifts up. So a little bit less harmful to our cells. Some diseases that can arise from a frame shift mutation are things like sickle cell anemia. So the protein that makes the red blood cells got messed up from a mutation, so they can no longer produce red blood cells properly. Tay-Sachs or color blindness. So again, color blindness would be like the protein in your eye that we use in our rods and cones. is messed up, we can't produce it properly, so you can't see color. That would be an example of a non-frame shift. So just that one specific gene is not working properly, but a person with color blindness can still make insulin and all of the enzymes and all of the other hormones, all of the other proteins are still functioning properly. Just that one thing isn't working in their body. So that's why it's not the end of the world, okay, but it still isn't great. Okay, not as bad as frame shift, but still not great. So yeah, that is basically all I had to talk about for this. Hopefully it makes sense, but if not, please feel free to ask me any questions. Talk to you later. Bye.